Welcome back to Biology Boost Up. Today in this video tutorial session, our topic of discussion is all about the next part of gene regulation. And at the end of this video session, I talking about how the presence or absence of lactose and glucose control the whole gene regulation system or the Lac Operon system. So now let's start. At first, what is Lac repressor? The Lac repressor is a protein that represses or inhibits the transcription of Lac Operon. It does this by binding to the operator which partially overlap with the promoter. And when bound, the Lac repressor gets in RNA polymerase way and keeps it from transcribing the operon. Now the questions arises here that where does the Lac repressor comes from? Now look, when lactose is not available, the Lac repressor binds tightly to the operator and preventing the transcription by RNA polymerase. However, when lactose is present, the Lac repressor loses its ability to bind with DNA and then it floats off the operator and cleaning the way for RNA polymerase to transcribe the operon. Now this kind of change in the Lac repressor is caused by the small molecule that is allolactose, an isomer of lactose. When lactose is available, some molecules which will be converted to allolactose inside the cell. Allolactose now binds to the Lac repressor and makes it change in shape so it cannot longer bind with DNA. And the allolactose is an example of an inducer, a small molecule that triggers the expression of genes. The Lac operon is considered an inducible operon because of it is usually turned off or repressed but can be turned on in the presence of the lactose or allolactose like a inducer. Now next, what is catabolite activator protein or CAP? When lactose is present, the Lac repressor lost what it happened DNA here. Binding ability. Now look, this RNA polymerase the way for RNA polymerase does not bind very well promoter and the transcribe to the Lac operon. That sounds like the end of transcript, but story it right won't do much more unless it gets extra help from catabolite activator protein. Now the CAP binds to a region of DNA just before the Lac operon promoter and helps the RNA polymerase attached to the promoter driving higher level of transcription. The CAP is not always active. Instead, it is regulated by a small molecule that is called cyclic AMP or CAMP. Now this CAMP is a hunger signal made by E. coli when glucose level are low. The CAMP binds to CAP, changing its shape and making it able to bind with DNA and promotes the transcription. Without CAMP, the CAP cannot bind with DNA and it then remain in inactive state. Now the questions here, how is CAMP made and how does it report glucose level? The CAP is only active when glucose levels are low, and then CAMP levels are high. Thus, the Lac Operon can only be transcribed at higher levels when glucose is absent. This strategy ensures that bacteria only turn on the Lac Operon and start using lactose after they have used up all of the preferred energy source, that is glucose. Now let's talk about some special conditions regarding Lac Operon or how the glucose and lactose control the operon system. So now let's start. First condition, when glucose is present and lactose is absent, then no transcription of Lac Operon occurs because the Lac repressor remains bound to the operator and prevents the transcription by RNA polymerase. Also, CAMP levels are low because glucose levels are high. So, CAP is inactive and cannot bind with DNA. Now, next, when glucose and lactose both are present, low levels transcription of Lac Operon occurs. The Lac repressor is released from the operator because the inducer, that is allolactose, is present. The CAMP levels, however, are low because glucose is present. Thus, CAP remains inactive and cannot bind to DNA. So, transcription only occurs at a low level. Next condition, when glucose is absent and lactose also absent. Here, no transcription of Lac Operon occurs. The CAMP levels are high because glucose levels are low. So, CAP is active and will be bound to the DNA. However, the Lac repressor will also be bound to the operator due to absence of the inducer, that is lactose, acting as a roadblocker to RNA polymerase and preventing the transcription of Lac Operon genes. Next condition is when glucose is absent but lactose is present. Here, strong transcription of Lac Operon occurs. The Lac repressor is released from the operator because the inducer is present here, and CAMP levels are high because glucose is absent also here. So now the CAP is active and bound to the DNA. This CAP helps to RNA polymerase bind to the promoter, permitting higher levels of transcription of Lac Operon genes. Thanks for watching my video. Please like, share and subscribe the channel. Thank you.